by WCHRI, and we will be doing some seminars in collaboration with the faculty, of which John Bell, it sounds like, will be speaking next month. And I think part of what we're hoping to get across here is both to provide some understanding as to what is precision health or what is precision medicine, and they're probably not the same thing. And I believe Dr. Canoe is going to help us out with these definitions, but also showcase the local breadth and depth of people operating in this space. And hopefully those who are interested and are coming or have come today to find out a bit more may realize that you're already working in this area without knowing it, as it's a newer concept and definition. I'm Todd Alexander. I'm one of the leads of WCHRI. I'm the associate director officially. And I'm trying to spearhead WCHRI, or the Child Health and Women and Children's Health approach to this area. I think it's important, it's essential actually, that we acknowledge that the University of Alberta is situated on Treaty 6 territory, the traditional lands of First Nations and Métis people. And I think within the context of precision health, and as healthcare deliverers, we also need to consider what the terms of that treaty stipulate. And one important term is that the government of Canada agreed at the time to provide a medicine chest to the Indigenous persons residing within Treaty 6 territory. And I think precision health is one of the ways that we can improve our ability to provide that medicine chest or care to Indigenous persons by taking their social, cultural, and genetic, unique genetic characteristics into consideration when we're providing healthcare to these populations, which many of us do here. I also think it's essential to thank WCHRI's funding partners, which is the Stollery Children's Hospital Foundation and the Lois Hole Hospital for Women, for all that they support, in particular the seminar series going like today and going forward. And for those of you who have a problem with recordings, today's going to be recorded, so just so that you know. And then without further ado, it's my privilege to introduce Dr. Peter Canoe, who is a recent recruit to the University of Alberta, right at the best month ever, March 2020 in Canada, which is obviously when COVID lockdown occurred. So he's a native of New Zealand, where he did his medical training. He then moved to Australia for the sports, obviously, but where he also did some clinical genetics training at Melbourne, the Royal Children's Hospital there. He also did a PhD in cell matrix biology in Melbourne before working in Australia at the Victoria Clinical Genetics program, and then being recruited to Queen's University in Ontario, after which he was recruited to the Hospital for Sick Children, all of where his expertise in skeletal dysplasias, both research and clinical expertise, was sought after. And then finally, landing here in the middle of a pandemic. And I've had the pleasure of working with Peter over the last several months and trying to drive some of our strategic healthcare precision health initiatives forward. And without taking up more of his time, I'd like to give the floor, or the virtual floor, to Peter. Thank you for agreeing to do this, Peter.
Thanks very much, Todd. I just want to clarify that everyone can see my screen. So, thank you. Kind introduction, Todd. I wish you had told me that I needed to wear a tie for this event, so I would have been appropriately dressed otherwise. But I'm really excited to talk about precision health today. It's something that is quite dear to my heart. It is, something that actually has brought me to Alberta simply because I was excited to hear about the province's pivot and what we can do in terms of precision health in Alberta. I'm just having some problems with moving my slides, so that's why I've just gone out to the slide share. So I want to start off by just defining the objectives for the talk, which will be a definition of precision health and talking about genomic medicine in particular in this area of precision health, and using some examples from genetic screening, diagnosis, and treatment, finally ending on the topic of big data and what that means for us. So for those of us who are not familiar with the terminology, there is a difference between the words precision health and precision medicine. And I'd like to say that a lot of what I'm talking about today deals with precision medicine. So precision health really is this approach of using the latest in technology to understand a particular condition, diagnose and treat a disease. So it encompasses much more than medicine because it encompasses strategic innovations in artificial intelligence, some of which we may actually use in precision medicine, but not completely so. And I think that's because many of us in medicine don't understand these, how these developments can actually improve our clinical practice. When you look back at genomic medicine and precision medicine, you can see there's been a steady build in our knowledge way back from the 1890s, when we were first starting to understand the building blocks in our body, culminating in the discovery of the genetic code by Watson and Crick, and the subsequent advances in technology that have occurred since then. And I would like to say that every decade that we go through in genetic medicine really sees an explosion in the discoveries, and that simply because the technology, the backbone or the platform, the technology is there, and that helps spur future developments as the decades go by. And so we are seeing progress that occurs at an exhilarating rate, even though it may seem, very slow to many of us who are in this particular field. So what's driving all these developments in genomic medicine? And I think the real big, poster child for the last decade has been the developments in high throughput sequencing that we all know as next generation sequencing, and the power that that has unleashed in terms of diagnosis for our patients with genetic disease. What we're seeing emerging now is, as we accumulate a lot of this information, is our ability to look at very big picture cohorts of data and mine that data even more in depth to identify clues that perhaps we can't actually, or we wouldn't have appreciated without these sort of big data analysis tools. And so that brings in the power of our computations, these computers that with multi-core, we're talking about 20, 30 cores now, and probably going up to 50, which are able to bring all these data sets in and mine the data and triangulate findings that we could not otherwise anticipate. We're seeing explosions in genome engineering. So the last decade has also seen the advent of CRISPR-Cas9 tools, which are now being perfected. So we're going on to base repairs and many other more efficient gene editing strategies that are enabling new drugs and new ways to treat people as well. And then there's also the artificial intelligence and the machine learning world, which I do not profess to know much about. I think the declining costs of sequencing, and we all know this, every decade it seems to get a lot cheaper, really fueling our ability to sequence more and more people using exome and genome sequencing. And then we're starting to need to think about, where do we store all this data, or should we even store the data? Because we're talking about mega loads of data, and it gets quite expensive to store data. And so one asks the question, perhaps the best place for DNA is to be in the body, in a cell. And if one needs to resequence the DNA, it's much cheaper to do that rather than just to store that. And so with the decreasing sequencing costs, we're looking at our ability to perform population-based sequencing. Would like to think that in the future, every patient that visits our hospitals, the University Hospital, the Lewis Hall, and the Royal Alexander, and the Stollery, would be able to benefit from sequencing and unleash the power that it can have. And of course, governments are also helping, with making funding available through Genome Canada, Genome Alberta, for us to pursue these endeavors. I'm going to compartmentalize our approach to personalized medicine in genomics, and to talk about, and use examples from genetic screening, diagnosis, treatment, research, and use all this under the umbrella of education, because I think it's really important as an academic center that we remember that our trainees are the leaders of tomorrow, and we need to be investing in their future and teaching them these new techniques as they develop. So what is genetic screening? I think that genetic screening is an example of a public health program that is typically, devoted to a particular population of asymptomatic individuals. And this provides provides us an ability to identify in this cohort individuals who are at high risk, who may benefit from prevention or early treatment. The best example of this is the Guthrie card that we're probably all familiar with. The Guthrie card was devised by Robert Guthrie in Buffalo, New York back in 1962 and rapidly spread to the rest of the world. And here's a picture of a baby here having their either their heel prick or probably their hand here, where blood spots are drawn and screened for several diseases, including the original that Guthrie suggested, which is PKU. So PKU is an inherited metabolic disease. It involves a deficiency of a particular enzyme, and that deficiency leads to an inability to break down this molecule phenylalanine, and phenylalanine then accumulates in the body and results in toxic complications, particularly to the developing brain. So individuals who have this deficiency should avoid foods that are high in concentrations of phenylalanine, and therefore we can see that if you can screen and diagnose this condition as soon as a newborn is born, you can then intervene by dietary means to prevent the complications on the developing brain. So screening tests, when one is thinking about a screening test, not every condition is suitable for screening, because there needs to be an evidence of a benefit or a risk or in terms of the screening. So, we won't screen for conditions for simply which we cannot do anything about. So some of the neurological conditions have no treatment. And so one may say, why screen? Others may argue that knowledge is empowering and people will want to know regardless of whether anything can be done or not. But we tend to stick to these main 10 tenets of screening here to think about when a condition is amenable to being screened. And most of all, it needs to have some sort of early phase that you can identify a patient before which they get sick, and during which you can actually devise some sort of intervention to prevent the complications that occur. So when we talk about screening, we can also talk about screening that occurs before birth and screening that occurs after birth. So screening before birth, I think one of the recent developments has been the use of this non-invasive test where a pregnant woman has a sample of blood drawn and from which fetal cells are extracted. And then genetic testing is undertaken. So we're seeing this, the emergence of this technology, and I would like to hazard a guess that, at some time in the future, invasive tests such as amniocentesis and chorionic villus sampling will simply disappear, because we will have the ability to make the diagnosis just on the diagnosis of a fetal abnormality just on the fetal DNA that is circulating. Right now, we have issues with false positive and false negative results, and therefore any false positive result may need to be confirmed by a more direct test. So there's been a lot of work done in this area to try and develop tests that are more sensitive, and currently people are looking at trophoblasts. So these are cells that are present during the development of the fetus and the placenta that also circulate in the pregnant woman's bloodstream. And you can actually sort for these cells using magnetic activated a method where antibodies are attached to magnetic beads to grab hold of the fetal cells with the corresponding antigen. And this picture here just shows a color karyotype of what could be done once you obtain the fetal cells from the pregnant woman. So why should we think about performing, genetic screening in pregnancy? And so this is a graph showing some EU numbers, and I just want to highlight or draw your attention to the last two bars, which are on the right-hand side of the screen, showing the current situation where there's the cost of the test. And if we were to use this fetal DNA screening on a large scale, and you can see the difference there in terms of the cost that would be saved, not to mention the reduction in the risk of miscarriage, which, occurs in any invasive test, such as an amniocentesis or a coronary venous sampling, although those are small. So when we look at genetic screening after birth, we look at conditions that, we screen for right now. For example, cystic fibrosis. I just put here that Alberta was one of the first provinces in Canada to introduce this. So with cystic fibrosis screening, so cystic fibrosis is an autosomal recessive disorder, which results in problems affecting the pancreas and lungs, leading to much morbidity and early mortality. We currently use this two-tier algorithm, which involves first a screen of a particular biochemical, followed by, if that was positive, followed by molecular analysis of the common CF mutations. So what are the other conditions that, we could consider screening for? So there's so many of them, and I've just put some examples out here now. We could think of Duchenne's muscular dystrophy that affects 1 in 3,500 males. There's no cure right now, though, this may be changing and may be emerging. And so one would argue, why should we screen for this condition? On the other hand, any parent of a son who's told you about the 2 years it took to make the diagnosis when they first brought this to the attention of the doc, because the kid was toe walking and nothing was really found, and so diagnosis took a long time, they would say, well, of course I would have wanted to know whether my child was affected by this. But the counter argument is, nothing could be done. There's no treatment available for this. Fragile X is another condition that one could screen for. This is one of the most common causes of intellectual disability in boys, with a frequency of 1 in 4,000. And now I'd like to highlight the case of spinal muscular atrophy. So this is a condition that we do not screen for right now, but for one where the landscape has changed with the availability of treatment. So spinal muscular atrophy, just a brief sort of dig into what the different phenotypes are of this condition. It's a genetic condition that ranges from a very severe presentation at birth with an infant or neonate not able to move and very floppy and hypertonic, to the more mild cases that can appear in adulthood here. And there's a diagnostic spectrum based on type 0 to type 4. So spinal muscular atrophy really is due to this particular spinal survival motor neuron basically dying early. And when these neurons die, they're not replaced. And this is what results in the neurological phenotype. So spinal muscular atrophy is another autosomal recessive condition, just like cystic fibrosis. So the interesting thing about this particular condition is that spinal muscular atrophy is due to problems in the survival motor neuron gene. And the body has done a curious trick during evolution. So it's got the normal copy of SMN1, and then it's also got the pseudogene SMN2. We often think of pseudogenes as these genes that are either on their way out or either way in evolution. But the really interesting thing about this particular condition is that I've just shown here in an unaffected individual, you get normal splicing of the gene and the normal gene product, but in SMA, you get a problem with a change there. You can see the base pair change at C to T between exon 7 and 8. And this leads to abnormal splicing and an abnormal gene product. And what happens there is that when you have less copies of SMN2, this influences the severity of the phenotype. So when you look at SMA, there's been a significant progress in the treatments that have been developed for it. And one that's already on the market is a drug that is an antisense oligonucleotide gene therapy. So these ASOs are very small oligonucleotides, which are about 12 to 13 nucleotides in length. And then they bind to the RNA just by a traditional Watson and Crick model. And so when they bind to the RNA, they have the ability to modulate the expression of the RNA. And so I'm just showing here what happens. As you all know, DNA is converted to messenger RNA, and then it moves on to, as a message, out into the cytoplasm, where it then influences protein synthesis. And if you have the single-stranded right here in the middle of the diagram, the single-stranded ASO binding to the RNA, that can result in inhibition and modulate how this RNA molecule works. So people have exploited this particular situation here to try and repair the splicing defect that occurs in the SMN2 gene. And so this oligonucleotide binds to this area which is abnormally spliced, and then it inhibits the abnormal splicing, resulting in more product of the SMN2 gene, therefore reducing the severity of the phenotype. So this is a treatment that's already on. And now more recently, you've all probably heard in the news with the fund, people with the GoFundMe calls in the newspaper asking for millions of dollars for this particular drug, Zolgensma, that has just been approved by Health Canada. And this is basically a gene therapy product which actually replaces the function of the non-working gene and restores the ability of the spinal motor neuron. And so Zolgensma is, as I said, just been approved by Health Canada for the treatment of SMA. The results look really amazing in terms of what it can do for a neonate who's floppy, and is probably going to succumb to a respiratory problem without any treatment. And so this, then begs the question, if we do have treatment for this condition, should we be screening for it early? Most of the clinicians will know that it can sometimes be difficult to make the diagnosis of SMA. In retrospect, everyone will say, oh yeah, of course, that's what the child had. But when we see the initial presentation of a floppy child, it may take some time for the diagnosis to be made. And in that time, the child is losing spinal motor neurons, which cannot be replaced. And the earlier you treat, the better the long-term prognosis. So that's this screening for this particular condition is something that the Department of Medical Genetics here is looking for. They're trying to develop an appropriate test, and so that we can bring it out for the province in due time. So the only other thing to say is that screening for SMA is already out in Ontario. They had a pilot project which demonstrated the success. And so speaking to the physicians over there, the issues have changed from can we make the diagnosis early enough to how can we get the patient this very expensive treatment, right? So the discussion items change, and all these medications and enzyme replacement therapies in particular are going to cost millions of dollars. And what we really need to have a discussion is how are we going to have a rational way of paying for all these drugs so that everyone can benefit, or what is our strategy, right? So I won't touch more on this slide because I've already highlighted the benefits of screening for SMA. Just to point out here that in genetics, we always consider that some people do not want to know, and we need to respect their right to do so. Moving on now to talk about genetic diagnosis. So I think some of the really fascinating new diagnostic techniques are coming out are these ability to extract tumor cells from a patient. And from that, we can extract tumor DNA or tumor microRNA. And with that DNA, we can then provide a diagnosis for the the cancer, and we can then also consider what suitable treatments are available. And so we're very fortunate here to be one of the sites participating in a national program looking at the use of this technique specifically for people that are affected with neurofibromatosis type 1. As you know, as people with neurofibromatosis type 1 age, they are at an increased risk of cancer. Women with neurofibromatosis type 1 face a 5 times increased risk of breast cancer, and so they're eligible for high-risk screening. But we really can't predict who is going to get the tumor, and oftentimes when one gets a malignant peripheral nerve sheath tumor, it really is too late for anything to be done. So can we use some sort of modern method of making the diagnosis with this non-invasive technique? And as I've alluded to, liquid biopsies are also becoming more popular in the management of breast cancer, because rather than relying on a tissue biopsy, one may be able to get all the information they need about how the grade of the tumor, and also using the genetic pathways, consider the most optimal treatment for this particular cancer. So this is the sort of the decade that's coming. We're going to be seeing these techniques more and more employed in routine clinical practice. So what other aspects of genetic diagnosis are there? And I want to go and concentrate on something that is very dear to the heart of clinical geneticists, and this is improving our diagnostic ability to detect genetic disorders in children and individuals. As you know, genetic disorders typically manifest in childhood, and so children are disproportionately represented in genetic disease. And many of these children, you know, before a diagnosis is made, they circle the system, right? They see multiple health practitioners. They also hospitalize multiple times because people do not know what's going on, and all this incurs health costs, right? And so we want to be able to refine further our diagnostic abilities and improve what we can do for these children. So in Alberta alone, we've got about 400,000 people who are affected by genetic diseases, about 7,000 different genetic diseases. And so no one physician could really, you know, say that they know all these conditions or can recognize the phenotypes and make that diagnosis quickly. So we need some sort of better way of making the diagnosis, right? We can't rely on the physical phenotype because the phenotype is so heterogeneous. It may just represent, you know, any number of 5 different conditions on our differential diagnosis. So back in, you know, the late 2008, 2009, very much so in the last decade, we've seen the rise of this agnostic diagnostic approach called exome sequencing, where we say a patient has a particular genetic disorder. We know they have one because of its early onset, the occurrence in multiple affected family members, et cetera, and we say, well, we don't really know what the condition is, and so instead of targeting 1 or 5 or 10 genes at a time, we're going to look at all the genes in the body, right? We're going to look at all the genes in the body and look at the protein coding regions. And so that has been what we do when we send someone off for exome sequencing. And a little tweak on the technology in the last 5 years has been the availability of genome sequencing. So in addition to the exomes, which are the protein coding regions of the body, we now then read the introns and the areas which we forever thought didn't really mean very much. And that really has added maybe 5% or so onto our diagnostic hit rate. So not a great deal. And so 50 or 60% of the children, the people that we test for a genetic condition where we're very suspicious of one, they don't receive a positive test result. So we know they've got something genetic, but we just can't find it through exome or genome sequencing. And one of the main reasons this is so is that the DNA variants that are identified through genome sequencing cannot be classified properly into disease-causing or not, because there's simply a lack of functional data. So we need to shift the paradigm. We need to move beyond the DNA and to look at what else is happening in the genetic code. And I want to use the analogy here of a musical score. For any of us who play music, you know, you can read the notes and you can read the notes simply, but you can't really play them unless you know the timing, unless you know, have all the other annotations, like, you know, is a note a semiquaver, or is it been given a longer time? How is it supposed to be played? Is it supposed to be played softly? Is it supposed to be accentuated? So all that reading in the musical score, all that detail really is missing when we simply look at DNA. DNA is terribly static over time, right? It doesn't change. And so if we're only restricting our focus to DNA, we're going to be missing a lot of information. For example, we're missing gene expression, right? We don't know which genes have been switched on and which genes have been switched off. And we can only do this by analyzing the RNA. So this is the transcriptome, right? And studies have shown that RNA sequencing is able to provide more information on the DNA variants that we've found by telling us whether there's allele-specific expression patterns, whether there's aberrant splicing, or whether there's some sort of outlying in genetic expression, either over or under. And this can be useful then to inform the interpretation of the DNA variant. We can then also add a further tier on with metabolic profiling. So metabolic profiling really deals with the ability to look at the small molecules circulating around the body, and that may give us an idea of a particular disease signature. These small molecules may change as they circulate in the body with time. You know, they may go up, they may go down in disease. And so we want to try and figure out whether we can uncover a particular disease signature. And this is something that we're working on right now in medical genetics, to see whether we can put together some sort of multi-omics approach using RNA sequencing and metabolic profiling to help us find a disease-causing variant. And lastly, we need to really think about the protein. If you think back about, you know, when genomics really started, it started in biochemistry, and it started with the proteins, right? Protein structure was first resolved when we had all the clever techniques to look at proteins, and we have got a state-of-the-art Department of Biochemistry at the UFA that is highly regarded throughout Canada because of its work in protein structure. And so we need to really think about protein-protein interactions and post-translational modifications. And these are issues that simply cannot be addressed by looking at the genomic data, and use this information in concert to be able to triangulate a disease-causing variant. And I just put this workflow here that I've tapped from somewhere on the line, where, you know, this whole approach is going to require not only the clinician, who I call the master phenotyper. Clinicians are very good at bringing oranges to a researcher and not apples, and separating them out. And that's our skill in being able to phenotype. But we need the clever people in the laboratory to be able to help us with these complex analyses. And we need our basic scientists also to figure out model systems and very ingenious techniques to be able to resolve whether a particular DNA variant is disease causing or not. So this is going to be an effort that really brings us all together, I think. And I just want to use you know, an example from the rare disease clinic that we used to run at the Hospital for Sick Children, where I was based on, to illustrate all this coming together. So this is a child who is now about 8 years old, and I have looked after him since he was born, when he came with a phenotype that was characterized by extreme pre- and postnatal growth restriction. And despite many different tests that we performed on him, we couldn't find the cause for him, and we went to exome sequencing, and we didn't actually find his cause until a reanalysis was done some years later. And this was because he was a compound heterozygote. So he had two different variants in a gene called GINS3, and we weren't very good at that time in calling these compound hets. We were very good at calling homozygous variants, but not compound hits. And the reason that this particular gene sparked interest is because the genes proteins are very important in DNA replication. And so what they do is, along with the CMG helicase, they come along like a zipper, unzip the DNA, and then prime the DNA for DNA replication. So other components of the genes complex had already been described with pre and postnatal growth restriction conditions known as the Meyer-Gauland syndrome, but variants in genes 3 hadn't been. So what did we need to do? Typically in genetics, we put this information out on a program called Gene Matcher. So it used to be that, you know, you would go to a conference and you'd meet your colleague and say, hey, I've got a patient with this. And they'd say, oh yeah, I have one too. And so, you know, computers have really accelerated this progress, because with Gene Matchmaker, you just type the coordinates in and provide as much or little information as you like, and then you get a hit. And we got a hit, surprisingly, with a researcher that was known to us very well, a researcher in Saudi Arabia, who was known to the Toronto team. But, you know, this would never have come up in discussions with him otherwise. You know, we've got so much else to talk about that he was sitting on another family here. And so P1 is the family in Toronto, and P3 are the Saudi family. As you can see, the Saudi family is consanguineous, and they have 3 affected children. And they had variants in genes 3 as well. So, you know, the first question was kind of answered. Do we have multiple families showing segregation? Yes, we do. But all the diehard geneticists will know that 2 families really is not enough. We need more than that. We also looked at the in silico prediction models to try and get an idea of whether the DNA variant was predicted to be pathogenic or not. And then we really needed to move to some sort of functional evidence, right? We needed to figure out in the laboratory whether this variant was doing something. And this is how I wanted to illustrate the fact that we need to be working very closely with our basic science colleagues to be able to get all this done. So this is work that was done in my lab at SickKids, and my postdoc, Kashif, has done a lot of this. The first thing that he showed was, because we were dealing with a very tiny child, we wanted to know whether the cells were smaller, and indeed there was no difference in cell size, and that shouldn't surprise many of us. As you know, the main difference between an elephant and a mouse in terms of cells is the number of cells, not the size of the cells, right? So the number of cells is less in the mouse than it is in the elephant, and we were able to show that although there were no differences in cell size here, our cells grew slower, suggesting that the DNA replication process was affected in some way. And these flow cytographs here just showing how the cells were stuck in a particular stage of the cell cycle, consistent with our hypothesis. And so we then went on to work further with the patient cells, because we wanted to identify whether there was a decrease in the protein, which is what we expected. And yes, we did. We showed that. And we thought then if the cells were proliferating at a slower stage, perhaps there was an activation of a replication stress response, which you would expect. So the body recognizes that replication is not occurring as it should, and it puts sort of checkpoints in place to try and rectify this. And this is what this evidence here is showing when we moved on to the cells. But human cells are difficult to work with, right? We were working with cells that had been that you can only keep for so long, because after passaging them in the lab for several passages, they basically lose their cell phenotype. And if you transform these cells using an EBV virus, you're also then inducing a change. And so you're not sure whether the readout is going to be an artifact secondary to the change. And so here we moved to work with a researcher in Montreal who actually studied yeast. And people will know that yeast are excellent models for cell replication. We were able to show some conservation in the domain, the DNA domain between humans and yeast in the area where we were looking. And we performed these similar experiments with yeast to show that the replication process was also affected in yeast, and to show that these yeast cells were also more likely to die when they were stressed. A lot more work has gone on. We've made a mouse model, and we've been doing a lot more work on replication speed, but I will not show it here simply because I wanted to use this as an example of how we can use science to bring things together. Lastly, we also looked at the protein-protein interactions. As you remember, these were missense mutations in the DNA, and so we postulated they weren't causing a splicing problem, they weren't causing haploinsufficiency, but what was occurring was a slight conformational change in the actual protein structure, resulting in changes in the interactions. And so here we used a clever technique called bioID, and this is where you label the proteins with a biotininated tab, and you can use that biotininated tab then to study via mass spectrometry how protein interactions are changed. And this is the wild type here, and just showing you changes that have been induced between the two variants that we've discovered. And so we can then figure out, well, what are these critical proteins that for which the interactions have changed, and how can that contribute to developing the mechanism behind this disease. The Undiagnosed Disease Program is really, really our flagship clinic in medical genetics that we hope that we can put patients through who have had exome and genome sequencing and have not been solved for these more clever techniques to be able to identify novel candidate genes. And we hope that that information will then feed on to our basic science colleagues who we will really need to rely on to help us obtain more functional data to be able to call a particular condition pathogenic. But also another outcome of this clinic is going to be our ability to enroll people into rare disease trials, right? So we not only want to be able to resolve a disease mechanism, that is obviously the first step in trying to think about a particular cure for a condition, but there may be cures out for some of these patients already that we can put into a clinical trial. And I put this slide here just to highlight the fact that if you look at, we all know about COVID right now and the COVID vaccine and the number of people that it's taken to be able to demonstrate one's side effects and tolerance and efficacy for these conditions. Now, for some rare diseases, you might only have, 1 in 2 million incidents, and you might have 20 people in Canada. So how on earth are we going to be able to move along here, these different phases that require tremendous number of participants? And I put this slide here to realize that we really need to reconsider how we perform trials for rare diseases, because the standard approach for many of the conditions that we already have trials for is not going to work when we're dealing with rare disease trials. And we need to be, we here at the university need to really reconsider how we do this, or we're going to get left behind with participating in rare disease trials, right? If we get bogged down in the requirements from the research ethics boards. So we would really like to, develop the University of Alberta as a rare disease trial center. We're already doing some of this here. And when I talk about rare disease trials, I'm talking about two aspects of rare disease trials. So one is repurposing old drugs, right? So if you have an FDA approved drug, right, you already know what the side effect profile is. It's already been trialed on many different people. It's much easier to repurpose that drug for the treatment. And so I just use as an example here a trial that I'm involved with for a rare condition called fibrosis progressiva. This is 1 in 1-point-something million people. And what happens is this is just inappropriate ossification of the skeleton as the patient ages. And the drug here was a drug called pelavarotene that had been put out by Roche many years ago for the use of chronic obstructive airways disease, which turned out to be something that can be used here for the treatment of fibrosis progressiva. And right now the trial has been successful, and they're looking at trying to get Health Canada approval for it. Another condition that for which an old drug is being used is Schmidt Metaphyseal Chondrodysplasia, where they're using the drug carbamazepine or Tegretol, which we all recognize to be an anticonvulsant. I do apologize that my talk is very bone-centric, and that's because much of my experience is in the world of bones. So what can we do about the new drugs? So, you know, there have been drugs now for hypophosphatasia, and what you'd recognize in this X-ray is basically there are no bones there. And so instead of producing bone, you don't produce any. And this is a defect in aspartase alpha, which is a particular part of the body where mineralization occurs on the cells, and there's enzyme replacement for it. Dr. Gerges in endocrinology and myself are involved in 2 different trials now for achondroplasia, looking at different agents to reduce complications. We've also been approached for a drug trial in Angelman syndrome, and I know that you know, the University of Alberta has always been very, very well known for its work on in eye genetics and ocular genetics, and the team here have been involved in developing treatments for blindness, and they've spoken many times in the newspaper about this. So coming along to now moving along from the trials to what are the other opportunities for us in genomic medicine? And as I've alluded to before, there's going to be the emergence of big data, right? There's going to be an emergence of all this health data that's coming along from all the various streams that we're currently employing. And there's going to be the ability for other clever people to come in and mine this data, to look for trends and to try and answer questions that, you know, us in genetics maybe have not even thought about because it's just not in our sphere of research. And so collaboration is the key here. I would like to suggest that the digitization of health information that we've seen in the last and this decade is going to be the single biggest mover for big data, right? And so what I mean by this is I put out here a standard medical note taken, you know, that's been probably with us for forever. I just put the time point 1900s there, but it's one that we use all the time, how we detail information. The problem with it is not searchable. And so what do? We employ an army of summer students to come along and comb the files to be able to extract anything meaningful from it. What we're moving to now with our EPIC or Connect Care system is our ability to codify clinical information. And once it's codified, it's actually searchable as well. And there's some improvements on that. We have a program that we will be using in genomic medicine called Phenotypes, which will give us the ability to actually codify genetic information. And so one can then search using terms like microcephaly or hypertelorism to pull out salient points. And moving on, we're now also at the cusp of using artificial intelligence to recognize syndromes, right? We know that this is being employed for screening, looking at screening COVID with it. They're screening for people at the border, but we also have programs, and this is the program called Face to Gene, where you upload a patient photo and then a list of syndromes comes back. Right now, you know, the programs are only as good as the information that have been fed to it, but they are going to get progressively better with time. And I've alluded to, you know, that I talked about gene matchmaking, our ability to reach out to others for help and to discuss the information at an accelerated scale. But another sort of twist on this concept is this ability to plug in variants that we identify into a database to help inform the classification of the variant. So what I want to draw you to this attention is that in this case, ID 123 here from Lab A, a heterozygous variant was identified in the BRCA2 gene, and they called this a variant of unknown significance. They didn't know what it meant. Now, when they put this and they uploaded this variant to the database, they also found these 3 other labs that had described this identical variant. And in this situation, the 3 patients all had breast cancer, but what it was found that in addition to this variant, they also had a second variant in the BRCA gene denotated, either the 1518, the 1408, or the 1943 variants. And it was later found that it was these variants that were pathogenic and the driver of breast cancer, and that the 1593 variant that had been identified was simply likely benign. And this information then allowed Lab A to reclassify that variant at great speed and put out a report to tell the physicians that, look, the genetic cause of the breast cancer cannot be resolved at this present time. So more, going on more about the big data, I just want to pull out some points here. A Stanford University white paper talked about the fact that 153 exabytes of healthcare data was produced in 2013, and this is has been expected to grow exponentially 48% a year to 2020. And so there's huge amounts of data on the market. And there's also a growing market for big data in healthcare, which some people estimate to be 69 billion in 2025, right? And currently we're struggling with our ability in how to aggregate this data, how to mine it, and also how to protect this data so that we protect confidentiality of our patients. So there are many, many different players for the big data. You'll recognize the ones that have already been in the system, Amazon, Google, and Apple. And in addition to that, we've got to think about the government and the regulation that the government imposes on us, right? When we're trying to play with this. And we need to also bear in mind that we need to think about issues around privacy to protect our patients and really think about who owns the data. We always talk about the fact that the patient owns the data, but there's also another opinion that it's the researcher who generates the data who owns it. And so I think these issues really are not resolved, and these are considerations that we need to be working at as an academic university. So I want to finish off here by just highlighting some of the challenges facing Alberta. And these are challenges that I have not thought of myself. These come from a white paper that was developed by the University of Calgary. And it points out that our innovation networks are somewhat fragmented with a lot of competition and not enough collaboration. There's also the lack of supports and commercialization channels. So when we do identify an idea where we think we can take further, we don't know how to approach these industry partners, and there's no one that can actually help us in terms of who to talk to or who to turn to. There's also the divide between what academia wants, what the government is going to be imposing on us as restrictions, what the funding agencies desire, which is usually open source data. So there's all this conflict going on here, which hasn't really been resolved. And the coordination of a provincial-wide strategy, right, for precision health still remains unclear with many participants, and the decision-making process hasn't been articulated right now. And so these are issues that we need to work for. We have one single genomics laboratory for the whole of Alberta. We have 2 genetic services. We've got 2 major academic universities. And so there's a lot of competition going on here in terms of what we're talking about. I want to highlight just before I close here that we will be celebrating Rare Diseases Day with the help of the Women's and Children's Research Institute. We're going to be hosting this on Friday the 26th. Rare Diseases Day is on the Sunday the 28th. We're doing a shortened performance this year via Zoom simply because of the pandemic. We've got many speakers that I've got here. Todd, who's going to be speaking about his work on rare diseases in the field of renal disease. Toshi, who has been a pioneer with antisense therapies. Lawrence talking about rare disease trials. Oksana Sukoleski, we're going to be using her experience in Huntington disease to talk to us about the whole journey from gene discovery to the therapies that are available now. Hallie and Allison will be talking about the Undiagnosed disease program. And lastly, I've also got someone from industry, from Alexion, Jason, to talk about the journey that happens once Health Canada approves a drug, because it's quite a convoluted process. So with that, I'll end, and I'll be very happy to take any questions that people have.
Thank you, Peter. So if you want to put it in chat, or if you just want to spit them out, I think just as a point of clarification, we talked about precision medicine, precision health, but personalized medicine versus precision medicine, do you think these are synonymous, or how would you distinguish them?
A good question, Todd. I haven't actually given that one too much of a thought, in terms of precision and personalized. I think they're trying to get at the same thing, but the term personalized really does put the patient at the forefront. One of the reasons that I don't like personalized medicine is I'm a firm believer that as a clinician, we have always practiced personalized medicine. And so the concept is not a new one for us, but the ability to use more precise diagnostic tools is what is novel. But that's just my take on things.
I actually very much agree with you. I think we've always done personalized and precise medicine. It's just becoming more precise, right? Like we've always considered multiple factors in the treatment of an individual. So it's personalized, and it's just with the more data and more abilities, it becomes more and more precise. Dr. Davidge, did you have a question? I see you're very eager there. No question. Lawrence, did you want to ask something? You have to unmute. You're both muted.
That's controlled by WCHRI. You can just unmute everybody now, John, as we ask questions. So I'll let Lawrence ask a question, but I just want to say thank you, Peter. That was fabulous. Learned a lot, and I think there's much more we can do here. But I'm going to hand this over to Lawrence, who, John, have you allowed everybody to unmute? I know.
Lawrence, you can talk.
Yeah, everyone can unmute.
There you
Sorry, I was
go.
trying to figure it out.
Thank you.
I'm now unmuted.
Oh, watch out.
It's unmuted.
Thank you, Peter. Great presentation, great overview. I'd be curious to get your thoughts on something that's sort of come across the desk at the Precision Health Initiative in the university, is the idea that genomic medicine will identify genomic transcripts, which then translate into proteins. But how are we using the same ideas around big data, computational opportunities to understand proteins, their structure and folding? And is that something that is going to be important as we try to advance the ideas that you proposed here?
So I think the ability to identify protein structure is important. And right now we have a good understanding of some proteins and we can predict that, but not all. And so we need more innovation in terms of that. So I know this because for the GINS protein that I was talking about, the structure was known. And so we could annotate that quite easily using a 3D construction with my collaborator who's a proteomics person. But for some of the other genes that I found, that structure was not known. And so we could not generate, sort of a computer prediction. So our ability, I use it in bioinformatics and genomics, but this computing technology applies not only to RNA, but it applies to metabolics, with the small molecules and proteins as well. And they all need to go in concert together, right? One of the problems I think we can see is that sometimes you don't even know the guy down the road is working on a protein. And so we need to be able to, communication is going to be so important to us. This has been flagged already as a problem, that there are too many players in the place. And so we want to make sure that we are nimble and we use our strategic advantage as a university where everyone is, within the same precinct to be able to feed on each other's ideas and move ahead. Synergistically feed on each other's. And so I think, there's been, the WCHRI has suggested that we need to build some sort of structure in place, some sort of hub, a genomics hub where we can posit all this information so that it's available to people that want to work on this. And so, if I'm in need of a protein researcher, I can then go to the hub and find out who is there. And I think that's one of the things that we really need, that we need to work on. And one of the easier things that we can work on, Lawrence. But yeah, definitely protein structure. It's same applies for small molecules. People don't know how to analyze them. They don't know how the signature works. And we need the clever people from science to help us in that endeavor.
And can I just chip in as a member of the Membrane Protein Disease Research Group? We have a very strong structural people here in the Department of Biochemistry that are doing crystallization. So not only are they good at they're good at actually doing crystal structures, cryo-EM and MR, but they're also good at modeling. So I think, as Peter said, this link from the genetics individuals on this call to these protein structures people is really going to be key in this area.
There's a question here from Dr. Eric Patton about, and he's asking about his work in exercise therapy, and he's trying to figure out who would benefit from an exercise program. Now, this, Eric, is something very dear to me because, you know of some kids that go to the gym and 1 month later they look like Arnold Schwarzenegger, right? And you may be going for 10 years and nothing happens to you, and you start to think, well, it must be something in my body. And so I would like to argue that if we were using, the technological readouts, which is where it would fit in with precision health, to be able to predict, which programs would benefit which people, that is precision health. But it's in terms of the readout, I think, and the innovation in terms of how we're going to decide the prediction, right? As opposed to just looking at the physical. We need to think about some clever biomarkers there. One thing that's always struck me is that being in medicine, or forever, we've only had ESR and CRP as, the readers of inflammation. And we all know how non-specific they are, but yet no one's come up with anything cleverer, right? And so we need to use our innovation here to think about better biomarkers to monitor disease, right? So that we can think about non-responders and people that are going to have nasty side effects early on using the clever technology that we have available.
In your experience, Peter, with competitions to fund this kind of research, I was wondering, is there a place for using just clinical predictors like clinical tests as opposed to genetic or blood markers or in the precision medicine, or are we too far removed from the precisions from genetics and proteomics?
The reason I suggest that you need to use the more sophisticated markers is the definition of that, that precision or that personalized health. Precision health has been really with the technology developments, right? The emphasis has been on the technology, and that's the only reason I say that, as opposed to what we've always done in trying to be personalized in our approach.
I think it's worth pointing out that Dr. Riche feels that in some cases that the clinical definition of precision health would be suffice. I think it depends on the call and the area. So this area is being defined currently, and I think there's some different disagreements. So Lawrence, do you want to comment on that?
Well, I agree with Peter. It's about the technology, but there are other technologies that allow for making more precise determinations about phenotype. And digital technology is certainly one of those. Understanding how people behave in their environments and what they're exposed to and foods and so on. I mean, so the idea that there are many inputs to making more precise care decisions, including socioeconomic factors and lifestyle factors, is front and center with what the university sees as the precision health concept. So, but I don't disagree with Peter that the focus is on the technology, and in this case, genomic technology is one of those very important aspects.
Peter, there's a couple other questions in the box if you wanted to, or I could.
I can't, could you, yeah, go on.
You want me to read them?
Yes, please, yeah.
Yeah. So somebody's is asking, I'm interested in knowing what you think about big data versus mining data, analysis data, and its impression on precision health or precision medicine.
Yeah, I guess Perhaps I don't understand the distinction between the big data and the mining of the data, because I think the importance comes from mining the data in terms of looking for trends or whatever we're trying to figure out. But you know, from a setting it up process, there are two separate issues, because with big data we need to think about storage and privacy and who owns the data and what sort of framework we're going to have as a university around that. And then the mining, the users that are going to come on. So perhaps I don't actually appreciate your question. I'm sorry.
Yeah, I'm trying to interpret. Are you still on? Do you want to? Oh, I see. Can you clarify a bit, Oscar?
I'll just go on to the other question, which is, can you speak to predicting and then preventing adverse health outcomes through the detection of epigenome? Yes. So you know, this is an excellent question, because when you think about adverse health outcomes, right, and you know that the DNA doesn't change, we always think, well, it's either an expression or it's epigenetic phenomena where they're post-translational modifications. And so I did neglect to talk about the epigenome and the work that is going on currently. But yes, that is an important field, Donna. And we're actually participating in a national epigenetic study right now. That is one that is being run out of the University of Western Ontario, or Western right now, looking specifically at the epigenome and its ability to provide diagnosis. There are other researchers that are looking at the epigenome to figure out whether those signatures can be used to predict particular conditions or diseases and how they would change with treatment.
And anybody who's reviewing for CHR, it feels like half the grants I'm reviewing are in the epigenome right now. So I think this is a very emerging area that shows great promise for not just diagnosing, but treating, as Donna pointed out. I think, well, we're almost 10 minutes past the hour. So did you see, John? I think we almost had 70 people. This has been phenomenal.
Yeah, we got up to 71 there.
This is phenomenal, the interest. Thank you again, Peter, very much for your presentation, which